Wish you a very good morning to all. So we'll continue with chapter number 2. Chapter number 2 is microorganisms. They are our friends also, they are our fuels. So here when we talk about the relationship between the friendship and enemy, so sometimes microorganisms are useful for us and sometimes they are harmful for us also. So that's why they are our friend also, they are our foe also. So here we are discussing the important topic, that is useful microorganisms, harmful microorganisms, how the microorganisms are harmful to us. They are causing different diseases, they are causing food poisoning also. And next topic will come for the discussion, that is food preservation, how to prevent the food from the spoiling. So that we will discuss. And different methods are there. And last topic, that is nitrogen cycle. So in that also different points are there, different topics are there. So that these are the major topics in today's discussion. So before that, I want to revise something regarding this microorganisms. So as you all know that microorganisms are very microscopic and that we are not able to see with our naked eyes. So for that we need microscope, and that the scientist who described the first cell, that is Antony van Leeuwenhoek. Okay, that you are aware. Then the branch of science study with the microorganism, that is a microbiology. The scientists who study the microorganism we call as a microbiologist. So these are the common things we discussed in our previous video. And another thing we discussed, that is the major 5 groups of classification of microorganism. So in that we had discussion about bacteria, viruses, protozoa, algae and fungi. So total 5. First bacteria, viruses, protozoa, algae and last is fungi. So these are the 5 major groups of microorganisms that we have discussed. And another thing we discussed in our previous video regarding the useful microorganisms. So microorganisms are useful to making the food items. Suppose you come on the page number 27 in the textbook. So you will get to know that the lactobacillus bacteria help to make curd from the milk. So that everyone aware about, everyone know about it. So lactobacillus bacteria help make milk into curd. So there is a conversion in the chemical processes. So here also you can see the right side, the casein coagulation, that is a clump formation. The curd it is a clump. It is not liquid part, it is a clump part, and it takes place only when the milk is acidic. So lactobacillus is what? Lactobacillus are acidic in nature. So then only it will convert into curd, and that's why you will see there is a Sorin test. Okay, so here the conversion there, milk plus curd, so some amount of curd you have to add into the milk, then only the process will start, then only the microorganisms lactobacillus will multiply. So here in the presence of some spoon of curd into the water, into the milk, so that make the condition acidic, and it will convert into formation of curd. So from the milk, and that is a casein coagulation, there is a clump formation. So this is the useful of microorganisms. As already we have discussed, there is a useful of microorganisms in the food items. Next is there is a tenderization of meat. Now here also tenderization of meat. This bacteria act upon the tough meat and make it soft. So this process is called tenderization. So that's why the meat is soft. Okay, meat is soft. And another method that we have already discussed, that is fermentation. So here fermentation is what? The sugar is converted to alcohol. That you are aware, sugar will convert into alcohol. So that is Sugar convert into alcohol. So that is fermentation. Sugar will convert into alcohol. That is called fermentation. And now in this, the important bacteria help. That is yeast. Yeast means they are fungi. So fungi help sugar to convert into alcohol. So that is a fermentation process, and different jelly-like substances. If you know the agar, then it obtained from the red algae, that is Gelidium, and then next is a Gracilaria, is used in the preparation of various food items like ice cream, jellies, and paddies also. Okay, so this way we are using this type of microorganisms for the food items. Next is the commercial use. Commercial use is very common because it is related to industrial process. So industrial process, most probably they are running with the fermentation process. So fermentation process, in that the process of conversion of sugar into alcohol. Sugar into alcohol with the help of yeast, and that is called fermentation. And the fermentation is given in fermentation process is given in 1857. 1857, the fermentation process is given. 1857, fermentation process by Louis Pasteur. By Louis Pasteur. So in 1857, the fermentation process is given by Louis Pasteur. And what is fermentation? The conversion of sugar into alcohol with the help of yeast, with the help of microbes yeast. That is called fermentation. Now some of the MCQs that also I will come with a PDF file, and that I will combine this video and screen recorder also. So whenever the exercise or regarding your chapter content, so that I will go through this slide. So in that you will get to know what are the topics we have discussed in this video, and quickly we will try to complete. So next is the medicinal use. Third use is medicinal use. So medicinal use means what? We are making antibodies. So best example we can take nowadays, that is corona vaccine. Okay, Covishield, then Pfizer, that is from Brazil. So different companies, they are making the different vaccines, and they are giving us immunity against coronavirus. So that's why this type of microorganisms are helpful to make antibodies, and different antibodies are there, means antibodies are different pathogens. So here the medicines produced by living organisms such a bacteria and fungi which kill or stop the growth of disease causing microorganisms, or we can say that pathogens that prevent that present in our body, that is called antibodies. So they are helping to improve our antibodies. So they are providing us immunity. So here different examples we can take for the antibodies. In that streptomycin, I am talking about page number 29. So streptomycin there, tetracycline there, aromycin there, and chloromycetin. So these are the different vaccines we are aware, and some of the common known antibodies which are made from the fungi and bacteria, that is Penicillium notatum, that is a fungus. Penicillium notatum, that is a fungus, and from that we are making vaccine, that is penicillin. Okay, penicillin they are making. So Penicillium notatum, and we are making vaccine, that is penicillium. So here these are the useful of microorganisms as far as medical use is concerned. So antibodies they are making. So and after that some precaution must be taken after having this antibodies. So precautions should be taken by using antibodies. So antibodies should be taken only on the active advice of the doctors. Unnecessary, we are not able to take any medicine. If you are having paracetamol every day, if your body temperature is normal, your body temperature is 37 degrees organs, it will damage to your liver. So that's why unnecessary taking antibiotics or medicine that harm to your body, it harm to your each and every part. Each and every part it will harm to the each and every important organs of the body, and that's why we must have to advise, we must have to consult with the doctors, and then only we will have this type of antibodies, means medicines. And we are not supposed to take wrong doses also. Sometimes if you know that continuous doses we are having, after 2 minutes we are taking another medicine, after 1 hour we are taking another medicine. So no, it is totally wrong. So wrong in the sense, each and every antibodies, each and every medicine, it will work, it will start its working up to some time. Suppose paracetamol there, it will continue its reaction, it continues its work up to 8 hours. So after 8 hours you are supposed to take another medicine. So there must be some dose. And production of vaccines, nowadays we are having Covishield, the vaccine, and that is very common, and all people are having, all people are taking also. So here the process of increasing the immunity, that is called vaccination. So next, vaccination process. So vaccination is what? Vaccination is it's a process in which we are improving our immunity. So we are getting some immunity from this vaccine. So here these microorganisms are used in the production of vaccines, which protect humans and other animals against several diseases by providing immunity against concerned disease. If any disease would be there, so we are taking any vaccine. So before any disease, suppose polio vaccine even coronavirus. Nowadays we are taking Covishield. Okay, so prevent that coronavirus. That prevent from coronavirus, then polio virus also prevent from polio. So that's why we are having this type of vaccines. And that process of giving vaccine to improve immunity, that is called vaccination. Next is the immunity. Immunity is what? It's a body ability of the body to fight against the microorganisms. So there is disease causing microorganism, that is called immunity. And each and every human being, each and every animal, each and every living organism, their immunity power is able to fight against. If the immunity power is not able to fight against the microorganisms, then we will be suffering from some of the disease, some of the illness. So it's better to improve our immunity. When you want to improve your immunity, you must have to take appropriate food. So then only you will be able to improve the immunity. And some common diseases nowadays we are suffering, that is diarrhea, cholera, then tuberculosis, after that malaria also, and polio, hepatitis B, tetanus, smallpox, prevented by the vaccination. So this type of dangerous disease can be prevented by the vaccination. So in future we must have to avoid, if you want to avoid this type of any disease, so we must take vaccines. So then only we are able to prevent such diseases. And for that, it's a schedule. There, government is also making one schedule at particular dose. After one dose, after how many days you are able to take dose. So that's why there is a schedule, and a particular vaccine is given at a particular age. This is called immunization. So 1 to 5 years, 5 to 18 years, 18 to 40 years, 40 to 60 years, 60 above, so their vaccine pattern is different. So in that the dose will decide how much dose they are able to take. So accordingly they are able to give vaccines. So this is about the immunization process. Next that is a blue-green algae. So here they are helping in the nitrogen fixation. So they are making their own food, atmospheric nitrogen they are converting. So this is commonly known as biological nitrogen fixations. That we will study in this cycle, the endo cycle, that all things we will study. Now cleaning the environment, another use. So microorganisms are useful to clean the environment. If something is lying, if some dead body is lying on the road, you know that that's very what we can say. If dead body is on the road, and that will be decomposed by the microorganisms. After 10 or 15 days, you will not see that the body is on the road. So simple. So in that they are helping to clean the environment. Some other uses of microbes, that is tanning, means to convert animal skin into leather. So in that way also these microorganisms are useful. Now here if we talk about bacteria live in the digestive system of the ruminants like cows and goats. The bacteria help in the ruminants to digest cellulose present in grass and plants. So here also bacteria in the ruminants, means cow, buffalo or any other animal. So when they are having grass or any plants, so in that cellulose is present. So bacteria will help them to digest the cellulose. In that way also these bacteria are useful. Now harmful microorganisms. So we are coming to the next topic, that is harmful microorganisms. So harmful microorganisms, generally microorganisms cause diseases first, and second is what? The microorganisms cause food poisoning. Diseases means what? If any microorganism will enter to our body. So that time, if our immune system is not powerful, if our immune system is not able to fight against microorganisms, then that microorganism will affect the particular organ, and then we are suffering from some of the diseases. So that's why we will see some of the symptoms also. Suppose I will give you a common example. If you are affected by virus, common cold, just take common cold virus. Okay, common cold. So in that, virus will enter the body. So that time our body is not able to fight against this viruses. So what will happen to the common cold? And it will be remain for 2 or 3 days if you don't have any vaccine, any medicine. So it is a replicated cycle in that viruses will multiply. Okay, so same way in bacteria also, like a cholera there, tuberculosis there, then diphtheria there, food poisoning and plague. So these are the diseases affected by the bacteria. Viruses we are aware. Suppose we can take this example of viruses, that is coronavirus. Nowadays this word is very famous, corona. This word is very famous, corona. So this coronavirus will enter to our body, then what will happen? If our immune system is not powerful, so they will affect to our respiratory organs. Respiratory organ means lungs. So this coronavirus directly affect to our lungs, and they stop the supply of oxygen. If the supply of oxygen will be stopped by this virus, then we are not able to breathe, and that is a breathing problem. And you know that there must be some percentage, the oxygen level must be more than 90. If it is less than 90, we can say that we suffer from some of the viruses, and commonly for the respiratory system we can say the coronavirus. Okay, so the oxygen level must be more than 90%. So there will be some effect, but we take care and we will able to fight against. Our immunity is like that; it can be fight against this virus. If we become careless, then this number will decrease, and when number will decrease, what will happen? You know that different species are available in our society also, in our village also, in our city, or all over the world. What is happening nowadays, you are aware. So this is also one of the big concern. And here in the human being, the microorganism called disease, if you call the disease causing microorganism called as pathogens. The disease causing microorganisms called pathogens. And here what is called communicable disease and what is called non-communicable disease. There are two types of disease. Very clear in 4th standard it was there. Communicable disease, non-communicable disease. Communicable disease means what? The disease will spread. Disease will spread from one person to another person. That is communicable disease. And non-communicable means the disease will not spread from one person to another person. So here common communicable diseases we can say that cholera there, common cold there, then chicken pox there, and another that is tuberculosis. These are the communicable diseases. Means it can affect another human. So viruses are there, bacteria, protozoa, fungi, and protozoa cause malaria, kala azar, amoebic dysentery, sleeping sickness. These are the diseases caused by protozoa, and malaria it is caused by, you know, that female Anopheles mosquito. Female Anopheles mosquito, that is for the malaria, and dengue, that is Aedes mosquito. Female mosquito, female Anopheles mosquito responsible for malaria, and Aedes mosquito that is responsible for dengue. Fungi in the ringworms means skin disease, and here you can see that there is a different modes of transmission of disease. So in that the disease will be transferred by water also, by air also, by contact, by handshaking, or in many means. If communicable disease would be there, it will spread anywhere. If I will sneeze, if I have common cold, if I sneeze, so that water droplets will come in contact with environment, and that water droplets it carry the micro pellets, okay, micronutrients. So they will enter to the body and they will cause disease. Next, the breathing problem related diseases, common cold, tuberculosis, and pneumonia. Okay, now in here also we can see corona disease. Next, with the help of water, drinking, when we are having spoiled water, or we can say dirty water, so thousands of bacteria will be there, and they are also causing some disease. So in that, most commonly the disease causing microorganisms which are present in the dirty water, so they cause diarrhea. And next is the true food we eat, that is protozoa is present, bacteria present, even the spore of fungi is also there. So that's why vegetables are good for our health. So that's why medicine, whichever Ayurveda there, Ayurvedic medicine, Ayurveda, the branch of science that deals with the medicines made from the fruits, plants, and the different vegetables. Okay, so that is good for us. And next is insects and animals. So in this we have to learn this word. These are vectors. Vectors. Okay, vectors means what? You can see on the page number 32. Vectors. The animal and the insect are called vectors. The vectors carry the disease-causing microorganisms from an infected person to healthy person and cause diseases. Very common. Suppose we'll take example of this one, coronavirus. Now one of the person is affected by corona, means case is positive. Now he will come in contact with you. So what will happen? That vectors, means disease causing microorganisms like virus, this coronavirus will enter to your body. And that's why we are using mask, we are using sanitizer, we are using different means to prevent from coronavirus. And you know that the vectors, we call it the spreaders. Vectors we call it the spreaders who spread the disease. Next, that is a housefly. So in that the cholera, typhoid, dysentery, tuberculosis are known spread by the houseflies. And the mosquito, that is you know that the mosquito is responsible, that is female Anopheles mosquito responsible for malaria, and this is transmitted by the plasmodium, that is protozoa into the human blood. The plasmodium causes malaria, and a fatal disease, the Aedes mosquito act like we can say the carrier of dengue virus. So this is about the vectors, and so you can see on the page number 32 also the housefly, female Anopheles mosquito there, that is responsible for the malaria. Then housefly, so they are responsible for the various diseases like a cholera, typhoid, dysentery, tuberculosis. Okay, so these are some diseases. Now, general preventive measures against microbial diseases. So here, you come on page number 33, you will get to know that the wash vegetables every day before cooking them, and isolation. Isolation is what? Isolation word we are using to get separate. If person is suffering from any deadly diseases, means communicable diseases, the person must be separated. Nowadays it's simple. Coronavirus. Some person is affected by coronavirus, corona positive. What we are doing? We are giving them separate rooms. Okay? So in that they are quarantined. The quarantine word here is means they will separate. Means they will isolate. And vaccination must be done. If possible, drink somewhat warm water, somewhat hot water, and maximum possible take care yourself. So these are some preventions. Now in the animals also some diseases are caused. So bacteria in cattle and in the poultry also. Viruses, same thing in the cattle and poultry. Then in the plants also some of the disease. In the rice, that is a bacteria. Wheat, that is fungi. Sugarcane, that is a fungi. Viruses, tobacco, you know that sometimes tobacco plant, it is getting affected, it become black because of virus. And that is bindi, that is okra, in that also virus. So next topic, that is cause food poisoning. The microorganism also cause food poisoning. Now food poisoning is what? The food gets spoiled. If you are keeping food outside for a long time, so what will happen? The food become poison, food become spoiled. So that's very danger if we are having this type of spoiled food. So we will vomit. We will do vomiting. So this is about the food poisoning, and try to see the definition of food poisoning. The microorganisms that grow on our food sometimes produce the toxic substances. I am writing the toxic substances. Toxic substances. Food poisoning, they are producing toxic substances. It means microorganisms are producing toxic substances or poisonous substances which enter to the body and harmful to our health. So the toxic substances produced by microorganisms make the food poisonous, causing serious illness. That is called food poisoning. That is called food poisoning. Main word is what? Toxic substances. Toxic substances. Now here some of the bacteria are there. So here you can see the bacteria causing food poisoning, that is Salmonella. Salmonella, you can see the figure also. Then Staphylococci and Clostridium. Staphylococci and Clostridium. And fungi causing food poisoning, that is Aspergillus and Penicillium. So these are the some agents, some disease causing microorganisms. They are producing toxic substances, and that's why the food become poisoning. And this process we call as food poisoning. Now another topic, very much important topic we are going to discuss, that is food preservation. Food preservation. In that we have first chemical method, we are using chemicals. Second, physical method. So in that chemical methods we are adding chemicals, and physical method we are using different instruments. And here one of the important method of food preservation, that is pasteurization, then dehydration also. So these are the some important techniques we are going to discuss. First you will see the food preservation. Food preservation is what? Food must be preserved to stop food poisoning, to avoid food poisoning. That's we call as food preservation. And that conditions required, if this type of conditions we want to stop, this food poisoning if we want to stop, we want to preserve, and preserve in such a way that conditions, first condition, the conditions are dry. If dry condition is there, the bacteria will not grow. Second is what? The temperature is very low. Third is what? The temperature is very high. So high and low temperature. So each and every microorganisms will die, will killed at a particular temperature. Okay? So and some bacteria will survive at any of the temperature. Suppose virus there, it can survive anywhere. It can survive at any temperature. So this type of microorganisms, first dry condition, increase and decrease temperature, means low and high temperature. Next, the definition of food preservation, you can see that the process by which the spoilage of food is prevented using the chemical or physical method. The spoilage of food will be prevented by chemical or physical method, that is called food preservation. That is called food preservation. The first chemical method we are having, that is food preservatives. Food preservatives, we are using different chemicals. Different chemicals you have seen that underlying part, that is sodium benzoate, citric acid, potassium metabisulfate. So these are the common food preservatives. And when we are using these common food preservatives, the microorganism growth will stop. So they are not able to spoil the food. So that's why we are using this type of chemical method. Another method we are using, that is common salt. NaCl we are using. Common salt. NaCl also we are using to prevent the food spoilage. So prevention of food can be done with the help of common salt also. Next is with the help of sugar, then oil and vinegar, then important thing, that is pasteurization. This topic, pasteurization. So it is related to the milk, Amul dairy, Amul, you know that each and every dairy they are following this method, pasteurization method. In this, what will happen? Thus, pasteurization is a process for preservation of milk. This is for the milk. The process is for the milk, pasteurization. So here the milk is heated about 70 degrees Celsius for 15 to 70 degrees Celsius for 15 to 30 seconds. 15 to 30 seconds. And then in this, at this stage, if any bacteria would be there, if we are giving heat, means we are boiling the water, boiling milk. So that time it will reach to the 70 degrees Celsius, means at that time if any bacteria will be there in the milk, they will die. They will kill. It will be killed. So this process we call as a pasteurization. And this is derived by Louis Pasteur. The name of the scientist who has given this method, that is Louis Pasteur. In which year? It is in the year 1862. In 1862, he has given this method, that is pasteurization. So Amul dairy, even different dairy, in the pouch you are getting milk. So that is followed by this method. They pass from this method, means milk must be heated at 70 degrees Celsius for 15 to 30 seconds. So at that time, if any bacteria or any microorganisms will be there in the milk, they will kill. Okay, so that method is called pasteurization. Next is a cooling or freezing. So here we are using refrigerator. So cooling and heater and refrigerator we are using. The canning or vacuum packing. So different packets are available. That chicks are available, different potato chicks and kurkure and that. So that is done with this method, vacuum packing. So nitrogen filled with that. Then prevention by heating. So it must be given at very high temperature. So temperature would be more than 135 degrees Celsius. More than 135 degrees Celsius. And this is called UTH. The process is called UTH. So it is UHT. Sorry, it is UHT. It is UHT. Ultra heat treatment. It is called ultra heat treatment. It is called ultra heat treatment. Means any bacteria, most of the bacteria will die at this temperature, 135 degrees Celsius. Most of the bacteria, most of the fungi, most of the disease-causing microorganisms will die. So this process we call as ultra heat treatment. Next is what? Dehydration. Here dehydration is what? Water is removed. Removal of H2O. Removal of H2. H2 is water. So removal of water. So that is called dehydration. The process of removal of water from the substance is called dehydration. That is sun drying. The process is called sun drying. And dehydration in our body also, where we are doing too much work, we don't have, we will not drink water. So that time what will happen? Dehydration. So we are also experienced with the dehydration. So means water will remove. So but here in the sense it is for the food. So water will be removed under the sun. So that is called sun drying. Now last topic we are going to discuss the nitrogen cycle. Before that I want to go for the advantages of food preservation. So that you can see 4 advantages of food preservation, page number 36. It decreases the wastage of food by avoiding spoilage. Second, increase the storage period of food materials. Third, nutritional value of the food is retained for a long period. And ensure the food availability at distant places and during the off seasons also. So these are the advantages. Now advantages of food preservation in a particular season. Suppose we can take example mango or mango juice, orange juice, or any material you can take. introduced into the can. Now canning process, in that the food will be stored for a long time and it will be transferred to the different cellars. So here what will happen, the food will not lose its natural taste, okay, because we are passing with the food preservative, chemical, mechanical, physical method. So these are the advantages of food preservation. Now here the last topic we are discussing, that is nitrogen cycle. So here last topic we will discuss, that is nitrogen cycle, and already diagram is there in the textbook. So nitrogen cycle we are discussing now. So advantages of food preservation that we have completed. Now last topic we are going to. So here N2 cycle is nitrogen cycle. N2 is a nitrogen cycle, and it can be asked in exam. Draw a label diagram of nitrogen cycle, and this we have to write in the notebook also. So today our work is this: draw a label diagram of. So already those who have completed this work, don't worry, and it is there in the NCERT question also, so that can be done. So first step, second step, third step, fourth step, and the fifth step. So total 5 steps of nitrogen cycle. So first of all, what is nitrogen cycle? So nitrogen cycle is a cycle where the nitrogen is fixed and used by the plants and the animals, and later returning to the environment, to the atmosphere, and again come back to the plant. That is called nitrogen cycle. Means nitrogen will it is 78%. The maximum nitrogen in the atmosphere is 78%. So we are not able to take directly. So it will be transferred first into the plants or by animals, means another way, and again transfer back to the environment, back to the atmosphere. Means in the atmosphere it is there, taken by the plants, again back to the environment. It is a cycle. So we can say that nitrogen cycle is a cycle of transfer of nitrogen or cyclic process of nitrogen from atmosphere to plants and plants to atmosphere. That is called nitrogen fixation, nitrogen cycle. Now different steps of nitrogen cycle. 5 steps are there. N2 fixation. First is N2 fixation. N2 fixation. Second is nitrogen. N2 is nitrogen, and N2 assimilation. Nitrogen assimilation. Then ammonification. Then nitrification. And the last is denitrification. So these are the 5 steps of nitrogen cycle. First is nitrogen fixation, second is nitrogen assimilation, third is ammonification, fourth is nitrification, and the fifth is denitrification. So here, according to the different steps, first nitrogen will be fixed from the atmosphere to the plants. So here we know that nitrogen is 78% in the atmosphere. So here what will happen, the atmosphere, from the atmosphere, the conversion of free atmospheric nitrogen into nitrogenous compounds. So first it will fix into the nitrogenous compounds, that is called nitrogen fixation, and this can be done with the help of 2 methods. First is a lightning and second is a biological method. Biological nitrogen fixation. First lightning. lightning. Okay, so first is the lightning, and here what will happen, you can see the equations also. When lightning strikes, the nitrogen and oxygen of atmosphere react and form oxides of nitrogen. So N2 plus O2. So first I am taking this N2 plus O2. It will convert into nitrogen oxide. So it will form nitrogenous compounds. N2 oxygen, N2 nitrogen and oxygen combined to form nitrogenous compounds. Then it will form nitrogen dioxide. Then it will form nitric acid. And last it will form calcium nitrate. So this is a conversion by the help of lightning. Next is biological nitrogen fixation with the help of rhizobia bacteria. We know that in leguminous plants the bacteria are present. So they are converting nitrogen into converting the atmospheric nitrogen into the nitrogen compounds. So here some bacteria like cyanobacteria, nostoc and anamena are also fixed atmospheric nitrogen into nitrate. So nitrate will form at the end. So this all groups will form nitrogenous compounds. Next is the nitrogen assimilation. So process of conversion of inorganic nitrogen compounds into organic compounds that becomes a part of the living organism is called nitrogen assimilation. So it is just convert with the help of leguminous plants. So rhizobia bacteria are there, they are converting. So it is a nitrogen assimilation. So it is a conversion of inorganic nitrogen compounds to organic nitrogen compounds. Ammonification, so means ammonia form. When the plants and animals die, the proteins present in them are converted into ammonium compounds by the putrefying bacteria and fungi, and this process is called ammonification. And we can define the ammonification as a conversion of complex organic compounds like proteins into ammonia. So here ammonification is what? Formation of NH3 means ammonia. Last, that is nitrification, and this can be done with the help of nitrosomonas. Nitrosomonas name the bacteria. So nitrates are then converted into nitrites by nitrobacter bacteria, means we call as a nitrosomonas bacteria, and these bacteria are called nitrifying bacteria. So here nitrifying bacteria, nitrifying bacteria. So nitrification is done by nitrifying bacteria. Now last step is denitrification. Denitrification with the help of denitrifying bacteria. So in that Pseudomonas bacteria, they are denitrifying bacteria. The conversion of nitrates into nitrogen gas by denitrifying bacteria, that is Pseudomonas, is called denitrification. So here denitrifying bacteria, denitrifying bacteria. Okay, so this is the last topic what we have discussed, and already one diagram is there of endocycle. These are the 5 steps. 5 steps I can ask in exam. What are the 5 steps of nitrogen cycle, and draw a label diagram of nitrogen cycle. So this can be asked in exam. So first is nitrogen fixation, where the nitrogen will be fixed from atmosphere. Nitrogen assimilation, inorganic compounds to organic compounds. Ammonification, organic compounds converted into ammonia. Nitrifying bacteria, in that nitrosomonas means they are nitrifying bacteria. Denitrification means denitrifying bacteria, that is Pseudomonas, convert nitrates into nitrogen gas. So it is a cyclic process, conversion of nitrogen. So these are the topics in this chapter, and exercise already I have sent one PDF file in the groups. So in that in this PDF file, one part is already we have covered. So in PDF file, most of the students have sent their PDF file on this number also. So please WhatsApp on this number. So WhatsApp on this number, on my number. If you have completed your exercise, please submit. So this is my WhatsApp number. Please send all the PDF file on the WhatsApp number. So this is the end of the chapter. Those who have completed the exercise, please submit, and please make PDF file, then only submit. Don't take only images, because it's very difficult to assess, because all things must be recorded. So it's my humble request to make the PDF file. PDF file you will make. Okay? So then you will send on this number. So it will be recorded, and according you will get marks. So we have completed chapter 1 and 2. Chapter 1, crop production and management. Chapter number 2, that is microorganisms, friend and foe. Okay, so these two chapters we have completed. Next time we'll go for another chapter. So thank you. Have a nice day.